Transportation of food and minerals in plants. What do you mean by transportation? Transportation is a process of transporting water, minerals and food to all the parts of the plant body. Transportation. Transportation is carried out in plants by a system called vascular system. It is composed of conducting tissues or vascular tissues forming special tubes called xylem and phloem. Vascular tissues are the tissues that perform the function of transport of materials in plants. Now, vascular tissue are of two types, xylem and phloem. The xylem transports water and minerals in plants. The phloem transports food in plants. Xylem. Xylem is the vascular tissue extending throughout the length of the plant. It helps in the transport of water molecules and dissolved substances from the roots to the aerial parts of the plant. Now, the movement of water through xylem is one way. That is, the xylem conducts water only in the upward direction. The xylem consists of four components: tracheids, vessel elements, xylem fibers, and xylem parenchyma. Now, here are the different kinds of xylem cells. This is the tracheid. This one is the vessel. Here is the xylem parenchyma, and here is the xylem fiber. Tracheids are the elongated dead cells of xylem. Vessels are the elongated perforated cells. Perforated cells are the cells that contain pores. Xylem fibers are the non-living sclerenchyma cells. Xylem parenchyma is the only living component of xylem tissue. Phloem. Phloem conduct food from the leaves to all the other parts of the plants. They are made up of four components: seed tube, companion cells, phloem fibers, and phloem parenchyma. So this is the diagram of a phloem. Here is the companion cell with dense cytoplasm. This is the seed tube with seed plate. Now these are the parenchyma cells. These are the companion cells, and these are the seed tubes. The movement of food through phloem is two-way. That is, the phloem conducts food in both upward and downward direction. Absorption of water. Water is present between the soil particles. The root system in plants absorb water from the soil. So this is the diagram of a root cell. This is the epidermis of the root cell with the epidermal cells. Now this hair-like structure is the root hair. The root hair is bounded by an outer covering called the cell wall. Inside the cell wall, there is another membrane called the cell membrane. Now this space inside the root hair is the cytoplasm, and inside the root hair there is a dense body called the nucleus. Now the outermost layer of the cells in the root is called the epidermis. The fine hair-like structures that grow from the epidermis are called the root hairs. Now the root hair increases the surface area for absorption of water and minerals. Of water. The cells of the root hair have a cell membrane that allows water molecules to pass through but restricts the entry of larger molecules. Water moves across the semi-permeable membrane of the root hair cells through a process called osmosis. Now, semi-permeable membrane is a membrane that allows only certain molecules and ions to pass through them. So here is the diagram that explains how the water moves across the semi-permeable membrane of the root hair cells through the process of osmosis. Now, what is osmosis? Osmosis is the process where the water moves from a region of its higher concentration to a region of lower concentration through a semi-permeable membrane. So this is a diagram explaining osmosis. Now this is the outside of the cell and this one is the inside of the cell. Now outside of the cell has high water concentration and inside of the cell there is a low water concentration. As we know that in osmosis the water moves from high water concentration region to the low water concentration region through a membrane called semi-permeable membrane. Now experiment number 1. Now what is the aim of the experiment? The aim of the experiment is to observe osmosis in a plant cell. Materials required: resins, water and 2 glass bowls. Procedure. So the steps are as follows. Label the bowls as A and B. Bowl A and bowl B. Add some water to bowl B and keep the bowl A without water. Add a few resins to bowl A and B. Observe both the bowls after 2 to 3 hours. Observation. After 2 to 3 hours, you can observe that in bowl A, there is no change in the size of resins, but in bowl B, the resins have swelled up. Conclusion. So we can conclude that the concentration of water in bowl B is higher than the concentration of water inside the resins. Hence, the water from bowl B enters the resins through their coverings, which act as a semi-permeable membrane. Therefore, the water is absorbed by the resins through the process of osmosis. The bowl A did not have any water. Hence, the resins kept in it do not show any change in appearance. Now move on to experiment number 2. So the aim of the experiment is to demonstrate osmosis in a living plant cells through a potato osmoscope. Materials required: potato, knife or scalpel, concentrated sugar solution, red color, distilled water, beaker, pin and peeler. Procedure. So the steps are as follows. Peel off the skin of a potato of a medium or large size. Make a cavity in it with the help of knife. Clean a beaker and fill it 1/3 with water. Add 2 to 3 drops of color so that the water of the dish becomes colored. Fill the cavity of the tuber with concentrated sugar solution and keep it in the beaker. Insert a pin on the wall of the potato cavity to mark the original level of sugar solution in it. Leave the setup for about 1 hour. This is how the potato looks like. Observation. After an hour, you will observe that the level of the sugar solution inside the cavity of the potato has risen. So this is the beaker containing colored water. Now this is the potato with the cavity. This was the initial level of sugar solution. After an hour, the water moves into the potato cell and the level of sugar solution has risen up to this. So this is the final level of sugar solution. Inference. So from here we can conclude that the potato cavity is surrounded by living cells. The plasma membrane of each cell inside cell wall acts as a semi-permeable membrane. When the potato is placed in the beaker filled with water, water enters through the cell wall of the potato cells. That is, the water moves from its higher concentration to lower concentration. Therefore, the level of the sugar solution rises. Precautions. So the things that we have to keep in mind are as follows. The wall of the potato at the base should be thin. The potato should be kept vertically and use a fresh and recently peeled